Haere mai. It's such a pleasure to welcome everyone here tonight and to see such a fantastic crowd. Thank you for turning on the fine weather for us as well tonight, which has been awesome. It's amazing to be in this beautiful place. Just to introduce myself first, I am Dr. Sarah Schoenberger, the Head of Research at the Neurological Foundation. So tonight, we want to celebrate how the Neurological Foundation has passed a really important milestone. Since we were founded in 1971, we've now funded more than $70 million worth of research, which is so exciting. You can clap if you want.
Very well.
We know that there is still so much to do in neuroscience and neurology, but tonight we really want to celebrate all the progress that's been made, both in the lab and in clinical treatments. And I just want to say a special thank you to our supporters who've enabled us to do this over the last 50 years. We've always wanted to fund the best people doing the best research, and so I'm so excited to have one of those people here tonight. Distinguished Professor Sir Richard Faull. He's a distinguished professor at the University of Auckland. He founded the Centre for Brain Research and the Neurological Foundation Human Brain Bank. He's been awarded more than $65 million in research grants. And when we add in the philanthropic funding that the Centre for Brain Research has brought in, Sir Richard has brought in more than $100 million worth of research funding. So I think that deserves a clap as well. Sir Richard is a patron of many charities, and he was also made a Knight Companion of the New Zealand Order of Merit in 2016 for services to medical research. He's dedicated his life to helping people right across Aotearoa, New Zealand. And he's very proud of his Taranaki origins. And and I'm sure many of you know he has whakapapa links here to Te Atiawa and Ngāti Rahiri. Welcome, Sir Richard.
Yeah, thank you.
I have a recent copy of his CV and it's 125 pages long, so it's quite the read. But what we want to talk tonight about is how the Neurological Foundation has funded small inchstones over the years that have turned into some great milestones. And I think Sir Richard's career is a great example of that. So I'd just like us to start tonight by hearing from you about how a boy from Taranaki ended up in neuroscience.
Okay. Kia ora, tēnā koutou katoa. So, yeah, first it's good to be home, you know. It's seeing Mount Taranaki. Well, I haven't actually seen it today, but and coming home, I mean, I've sort of just gone around and said hello to a lot of people who have come tonight, and pretty well half of you I'm sort of related to. I mean, put your hands up, those who are related to me. Yeah. And those who knew my mum and dad. Yeah. So, and of course, it's, you know, it's a, despite all the things that Sarah says there, I'm really just a Taranaki boy at heart. And when you got that stamped on your bum, that's there forever. So it's wonderful to be here. And also I'm told, so I'm Ngāti Te Atiawa, and this hotel, I'm told, is owned by Te Atiawa. So we're home, you know, so nāpai, thank you. So, yeah, Tikarangi. Well, Tikarangi was where I grew up, and probably I would think I'm the most privileged, us 5 boys, and we had our mum and dad there, we grew up there, my mum and dad had the general store. And growing up in a little country area was probably the best education we could have. The school was only a 3-room school, and we weren't, you know, there were no problems of bringing cell phones into the room or anything like that. You know, you just got on and did your job, and also was straight up and down, was either right or wrong. And my mum and dad always said, you know, look after the people, And so all of us 5 boys, we delivered the groceries to all the different families around the district of whānau, and so we knew everyone, and we all learnt the art of communication. We all learnt how to talk to people, and most importantly was imprinted on our brain was help people. Don't get full of yourself. All the stuff you've heard is very nice, but it's all about teamwork, and it's about getting on and never ever forgetting where you come from. So coming home like this is the most wonderful thing in the world, and meeting people like tonight and is just very special. So thank you for coming along. So talking about how did I get into brain research? Well, there was no real big plan in a way. I was a bit of a nerd because I just loved learning. I loved finding out how things worked. I teamed up with the electrician across the road from our shop, and I used to go over there nights during the week and the weekend because I always wanted to know how wirelesses worked, And he used to get these wirelesses from all the farmers, and he let me pull them apart. And then he said, well, we'll use those bits and pieces, and now I'll teach you how to make a wireless set. So that and learning how cars went, so I sort of always had a spirit of inquiry, sort of a curiosity, and going through school, I had a love for biology, and going through, when I went off to Waitara High School, biology, I loved biology because we did frog dissections. We were looking at what made mammals work, i.e. humans. I mean, we're mammals. And then got up to the 6th form, and there was only 13 of us in the 6th form, 3 of us doing science, and we had to sort of teach ourselves. So you actually learnt how to learn, and you had to love what you're doing. And that's what happened off to Newport Boys High School during the final year there, because Waitara didn't have a 7th form. Got thrown into a big class. It was a class of the top scholarship science people. There was 125 in the 7th form there, huge. And 25 of us got put into a class because I'd come ducks at Waitara. They put me into this class with all the other hot rods, and I thought, how am I going to cope? But, I found that because I had learned how to learn, I actually survived it, and I loved it. And then I went off so I decided to go into medicine because I wanted to know how the human body worked. It was sort of a natural concept. I love maths and physics, but the human body seemed to be the most incredible organ ever. And so I got into Otago Medical School and I should say that when I at the end of the year at Newport Boys High School, we all sat the scholarship class, and I was one of a few of them who we got a Taranaki scholarship. That meant I had funding for all my years at university. Mum and Dad, neither of them they didn't go to high school. We didn't have much money, so I had the funds, you see, to go off to follow my dreams. So I went to the Otago Medical School, and in the 3rd year, when you're doing medicine in the 3rd year, you get to look at the human brain for the first time. You dissect the body and see how the whole body works, but then in 3rd year you get to see the brain. And the brain, I was this naive young Taranaki guy looking at the brain, and beautiful folded brain, and I thought, how does that make us who we are? Everyone here tonight is a special person. You're unique. I met some of you a few years ago. We're the same person tonight, And you meet up with your friends and years later, but you've got a stamp, and that stamp is in your brain. Your brain is who you are, it's what you are, it gives you your vision for life. It's something incredibly special. You don't realise how special your brain is until something goes wrong with it. So I was so taken by the human brain that I took a year. I asked if I could take a year out from my medical studies and just we could do a research year if you really wanted to, and you'd done okay in the exams. So I took a year out and did brain research, and I researched on rats. Because you can't research on people because you can't. People don't like being Well, you know, you can't do something to the brain and then sort of kill them and look at the brain, right? You can do that with a rat, so we had to work on the rat, and we found a new pathway in what we call the basal ganglia. It's the part of the brain involved with Parkinson's and Huntington's disease. So right in that early stage, I just loved that part of the brain, the basal ganglia. And so published, out of that year, we published about 4 papers, and I saw the professor, I said, listen, can I stay and do a PhD? He said no. He said, go and finish medicine and go try neurology or neurosurgery, and if you still want to do research, come back then. Because he said having a medical degree and knowing about people and patients is going to be the most important thing for your career. So that's what I did. So I graduated in medicine at Otago, and graduated on Thursday, married my wife on Saturday, headed north, and then worked in neurosurgery for a while, and that was really interesting. But I realised that neurosurgeons can only do things on the outside of the brain, take a tumour out or take a blood or take a clot out. We couldn't change the inside, because we didn't know how it worked. So that's when I decided to go back and do a PhD on the brain. And from there, I did that at Auckland, and then went off to the United States, and I worked on this basal ganglia cerebellum, the parts of the brain which control movement. And the top people in the world who were working at that time, one was in San Francisco, Bill Mailer, working at a big NASA research institute where they were developing the Skylab, but he was looking at the brain to see how space travel affected astronauts. He was one top expert in the field I was in. The other world expert, really, which was a person that he had also worked with, was at MIT and Harvard and Boston. He was the guru of the brain, of the basal ganglia and the cerebellum. So I went and I learned at the hands of the master. And Diane and I spent 3.5 years there. And I was learning techniques to come back to New Zealand, you see. Towards the end of the time there, I had to return. My money ran out, and I was going to have to return to New Zealand. And so I wrote home to Dad and said, listen, Dad, I need to stay a little bit longer, because to finish learning this technique and tidy up my studies. And I said, do you know anyone who could actually help? You know, if there's any. So, you know what, he went off and talked to us. Well, my dad was a great talker. Anyway, he talked to some Te Aotearoa Kamatua. And I knew we had a Maori whakapapa, but I never really knew much of the details. And, you know, hadn't been on a marae, and so, but they said, yeah, we'll help him. We'll help him we'll give him $1,000. And so Dad wrote back and said, oh, they want to give you $1,000. And I said, well, you know, I don't feel Māori. And I said, I don't know whether I can really accept that. And so he went and talked to them again, and then wrote to me, and of course it was snail mail, you know. And he said, you know what they said? They said, he's Māori, and one day he will come back and he will help our people. And that was so right. There's people here tonight, some Te Aotearoa people who I've worked with closely who are affected by a brain disease, you know, Huntington's disease, terrible disease. And so I come down here every year and so on. So you know, support not only from Taranaki Scholarships Trust Board for universities, but support from Ngāti Aotearoa, Te Aotearoa, Ngāti Rahiri for finishing the studies. And I came back and then got involved with all sorts of things at the University of Auckland, but slowly started to develop my brain research. So I think I'll probably stop there, because I know some other questions are going to come up, and if I keep talking forever, you all want to go home.
I think your story probably leads really nicely into the next question, because you had your family telling you it's all about people, and then you had your professor as well telling you to do your medical degree and learn about the people before you come back to research. So this year we have another important milestone, which is the 30th anniversary of the Neurological Foundation Human Brain Bank. And that's been all about the people right from the start and making those little inch stones into some great milestones. Do you want to tell us about that?
Okay. All right. So interesting, because when I came back from the United States, I set up my own little brain research group at the University of Auckland. It was just me, right? And again, working on rats, working on the part of the brain with the new techniques I'd learned to try and find out what controlled movement and what happened in the brain with disorders which we were trying to model, like Huntington's and Parkinson's disease. And so working on the basal ganglia. So after I was back for about a couple of years, I got some money from the Medical Research Council, and then I had a visitor come to see me, a professor of genetics. His name was Arthur Veal. Now, some of you here may realise that I was taught chemistry at, well, you won't realise that, but I was taught, the Veal family is a well-known Taranaki family. So Arthur Veal he taught chemistry at New Plymouth Boys High School, and he taught me in my final year there, and he was a wonderful guy. And a great teacher. Well, his son went into genetics and become a world-famous geneticist working with families in Huntington's disease. And he was at the Auckland Medical School. He came to see me. And he said, you know, Richard, he said, I'm looking after all these people, all these families, 400 families throughout Aotearoa, New Zealand, who have this terrible, tragic genetic brain disease called Huntington's disease. And he said, and this was the year 1980, and at this stage we knew it was caused by a gene, but we didn't know what the gene was, so you couldn't test for it, you see. And it was what's called a dominant gene. So if you really do have the gene in the family and your mum or dad have these symptoms, and if it's because of this gene, then 50% of your children will have a chance of inheriting it. So it was a diagnosis which was a stigma and a stamp on the family, you see. So he said, the families want someone, they know they can't test for the gene, because we don't know what the gene is, but he said, you know, if you look in the brain of their mum and dad after they've died, you'll be able to tell, and I knew I could tell, because the pathology in the brain is very characteristic of that disease. So you can look at the brain and you can say, yes, this person definitely did die of Huntington's, and that diagnosis is therefore correct. So he said, the families would like someone to check and look at the brain and see if of their mum and dad when they died, to see if they really do have this disease in the family. Would you do it? And I said, sure. I didn't know where this was leading, so he would arrive along every 3 months and bring a brain from a family in Hokitika, Invercargill, Christchurch, Northland or Auckland or wherever, and give me this credible, precious, you know, it's tapu, the brain's tapu from the family, and saying, look at this for the children. So we did. I got teamed up with a neuropathologist and we did a careful analysis. And so what was absolutely incredible was that the first few we looked at, we had to say it was positive, but those families said, well, listen, keep mum's brain, keep dad's brain, and you do research on it now, and you find out why the brain cells are dying and help our kids. And that was incredible. Well, we started, as the years went by, over about 10 years, we did special research studies using techniques I'd learned overseas, looking at chemical changes, cell changes. And what was the most interesting thing was that although it was clear they all had Huntington's, the ones we were looking at, there was variation between the brains. You know, there was the pattern of cell death in one brain was in the same general area in each of the brains, but the pattern was different, totally different. And so I thought, why is this? So I started talking to some of my colleagues at the medical school, especially in psychology, and they said, you know what you should do? You should go back and get the history from the family of the mum or dad who had died and find out what symptoms they had during life. What was their history? And I thought, what a great idea. So, and the psychologist joined us. They went out and they talked to all the families who had donated brains, and at this stage they're 35, and they got the histories, and the histories were different. Some of the patients had more mood problems, others had movement, whereas they had jerky movement, which is characteristic of Huntington's disease, but not all Huntington's disease, and some of them had mixed symptoms. So there were different variations. When we got that information, if you like, it's a track record of that person in life, and compared it with the variation of cell death in the brain, we found that people who had mainly movement disorders, they had lost cells in a very specific pattern. The ones with mood had lost cells in quite a different pattern. And the ones which were combined, they had combined patterns of loss. So what we discovered, I wish this was the idea I had, what we were able to find is we were able to use the brains from the family, from the whānau, together with the history, and we could look back and say which parts of the brain or the basal ganglia were involved with movement and which parts were involved with mood. And we could get fine with it. So although we couldn't do experiments on people, we were doing, these were like experiments, you know? So we found that in studying the human brain and knowing the pattern of the symptoms, and to know the pattern of the symptoms, we had to go back to the families for more and more information. Visit them 2 or 3 times, our psychologist did. We actually formed a relationship. When we went back to the families, we said, we need to know more information about your mum or dad. You know, they were overjoyed because they knew that we were getting every little bit of research on that brain. And so we were able to unravel, for the first time, what parts of the basal ganglia were involved with different types of functions. No one else in the world had done that. We published this paper, and it was a world-leading paper because the families had not only given us the brain, they gave us the story. And this was powerful. And you can only do that in a small country like Aotearoa, New Zealand, which is 4 million people, whatever, because everyone knows everyone else. You can ring them up and talk to them, and they come to, you know, so, and we go to a conference and talk about it, and people thought, God, this is incredible. And then big research groups overseas would start asking us, can you send us some tissue from families with Huntington's? And then we expanded and went to Parkinson's, and we started expanding our collection of tissue from the different diseases, because each disease was different because it involved different parts of the brain. So we were using the diseases and the human brain to unravel the function. We weren't just studying a rat. We were studying the Rolls Royce, you see. And it was amazing. It was amazing. So when we talked at conferences, they'd said, well, can you send us, could we do some of our studies instead of on the rat? Can we do it on some of your human brains from Huntington's or Parkinson's, looking at this or that? Because we've got techniques which you don't have in New Zealand, which we couldn't afford to have. So we ended up, we went back to families and said, are you happy to send a little bit of mum's brain to Oxford, to Cambridge, to New York, Zurich or whatever, to the top people in the world so they can do things we can't do here? And you know, the families were, in every single case, right up to now, they were overjoyed to think a bit of mum's brain is going off to Switzerland, going off to London, going off to Oxford or whatever. And one of them said, you know, it was like mum was getting the travel in life and death. They shouldn't get in life. Or else, you know, dad's living forever his brain, because it's being used by all these different people around the world. So I realised that we had the collection of brain, and I thought, well, you know, we should give it a name. We've really got a human brain bank of incredible tissue. Not only incredible tissue, but we know the track record of each of those people, you know, and that made us better than any other brain bank in the world. And it takes a lot of work to do that. And so in 1993, I actually went off to, I talked to Neurological Foundation because you see, we were having to pay staff off our research grants to look after the brain. And we didn't have much money. And so I said, and I just so happened that the medical director of the Neurological Foundation then was Philip Wrightson, who was a neurosurgeon who I had worked for as a young house surgeon, you see. So we knew each other well. And I said, Philip, you know, we've got a brain bank. If the Neurological Foundation could give us a bit of money to pay for a staff member, and then we'll put your name on it. We'll call it the Neurological Foundation Human Brain Bank. I didn't realise I was actually marketing, you know. Like my dad in the shop, you know. And so we took it to the council. Yep, so in 1993, we declared at that stage we had over 50 brains in the bank, and we called it the Neurological Foundation Human Brain Bank. So you say, so it's 30 years. So the brain bank actually started before that, but we didn't formally put our big label up until 1993, Neurological Foundation Human Brain Bank. And so, as you can see from the story, we were beginning, and I called it, we were doing brain banking the Kiwi way. You know, sort of following our nose, and no other brain bank in the world, you know, the United States have got a huge brain bank in New York. I know the director well, but their brain tissue is not nearly as valuable as ours because, of course, they get it from all over the United States. They don't keep contact with the families. And all they do is put the diagnosis, this is a Parkinson's brain, or this is a Huntington's brain, or an epilepsy brain, or whatever. They don't know the track record. And the other thing the donor families did when they registered to donate the brain of usually the person alive during life, we told them it's really important for us to get the brain soon after death, so they would organise it so that, and we'd work with mortuaries around the country so that we could get the brain as soon after death as possible, because that, before there's any degeneration going on, because it makes it less useful for research. So we get brains, you know, with what we call a short post-mortem time, a short time between death and when we receive it, of the order. The shortest is 2 hours, up to about 12, 15 hours. Now, compared to overseas, they usually go 24, 48 hours, you know. So we ended up having the best little brain bank, and we've ended up having the best little brain bank in the world. And over the years, so we called it the Neurological Foundation Human Brain Bank. As we've developed it over the 30 years, we've realised we could do a lot more. We could look at each of the brains and look at genes and do gene studies and use other fancy techniques, that we could learn more about that brain, and we needed more staff. So we go back to the foundation. And so now we've got 5 staff. So the Neurological Foundation in our book is number 1. You people here tonight, when you give money to the Neurological Foundation to support a cause like us, I mean, you are incredible. And, you know, in a country like Aotearoa, New Zealand, doing this and engaging with the community, you know, I was taught to engage in the community when I worked in the shop. All of us boys were. And they said, always give back, you know, always give back. So when we get additional results, we go back to the families and we tell them what we found, if there's something unusual or whatever, you know. So you end up never forgetting where you came from, never forgetting that the people who helped you do this research are the community. That's why I'm here tonight, actually. You know, we've had wonderful support from from people in Taranaki and of course my career and the whole works of it. So that's why I say don't, I hear my mum talking in my ear, I'll say, Mum, I just published this paper, got a prize or something, and she'd say, that's great, dear, now just get on with it. Get on with it. Don't forget what you're doing. And so, and that's, so this year we're going to celebrate, 30 years.
Do you know off the top of your head how many brains you've got in the brain bank now?
Well, we've got tissue from 1,000 brains. We can't just accept every brain because we can only accept brains that what we call have single pathology. If a person has Parkinson's and also gets Alzheimer's later in life, that becomes a complicated brain because you don't know what it's called. So we have to be very fussy, but the support we've got is huge. And not only do we collect donated brains from families, but also we've gradually got closer to the neurosurgeons because I did time in neurosurgery. I've always kept up with the neurologists and neurosurgeons in Auckland City Hospital. So there's a big neurosurgery unit there, a lot of neurologists. I got, 9 neurosurgeons now. So when any person comes to the neurosurgery for an operation, for a tumour or whatever, we work with the neurosurgeons and we get permission from the family and from the person who's having the operation if we can use some of that tissue for research. And that comes straight across the road to the medical school within 10 minutes. So we not only get brains from people when they die of a brain disease, we get tissue now also from people who go for neurosurgery at Auckland City Hospital. And so it's a partnership. So it's working with the doctors as well as working with the community and the families and the whānau, and the scientists, of course, and the as our research developed at Auckland University, we have found that we needed to get all the scientists to really work together. People who are doing anatomy and pathology like me, to people who are looking at the pharmacology, looking at culturing cells, other people doing physiology, all the different, and the people doing genetics, and even people looking at the effects of dance in the brain, all the rest of it. We thought, let's form a big club and get all the people who are working on the brain at the University of Auckland together, and let's give it a name. So we did this in 2009, called it the Centre for Brain Research. And that means everyone who's doing research on the brain, animal brain or human brain, are part of this club. And if you're part of the club, then you've got to share your results. So it's promoting collaboration. I didn't realise how powerful that would be, because when we launched this in 2009, we had 25 research groups, and today we're just doing our annual report. We've got 92 research groups. So it's amazing, isn't it? Working together as a team. I say, well, we're sort of the All Blacks of brain research in a way, because we're all different. And we just got to win the World Cup. That's the only problem. But it's just teamwork, and not getting full of yourself, not forgetting why you're there, not forgetting you're there to help people. And I was talking to a few people tonight who've been touched by different types of brain disease, neuropathies and all the rest of it. And I'm just so lucky that I go to work, I can choose what I want to do. I get paid to do the things I want to do, and how exciting it is when you get people to work together. And someone says, well, when are you going to retire? I said, why should I retire? When I get paid for what I want to do, I can decide what I want to do, and and it's not a job. It's a privilege. So I promised my wife Diana that probably in the next couple of years I am going to slow down, and I need to probably do that. Anyway, I'll probably talk a bit long on that question.
So this amazing collaboration, you're working with families, you're working with scientists, and you're working with the doctors and the surgeons as well. So what are some of the milestones that you've seen happening from all this collaboration?
Well, it's really interesting. When you plan your life, I often have graduate students coming and say, listen, I want to plan my career. And I say, well, just know what mountain you want to climb, but don't decide how you're going to climb it, and just keep your eye on that, and whatever opportunity comes up that's going to help you get there, just do it. Don't micromanage your career, and take every opportunity, and also take risks, right? Take risks, you've got to take risks. So one of the things we found, and this is from looking at the Huntington's brains, after we'd collected about 50 Huntington's brains, and the pathology was very clear-cut, but variable, as I've told you, we found that lying right in the middle of the brain, next door to the basal ganglia, in the middle of the brain, there's what we call a ventricle. It's a hollow part where the CSF is. And the lining round there is very distinct. It's called the lining of the ventricles. In rats and cats and monkeys, we've known that in that lining, there's still stem cells left over for when those animals were young. And those stem cells are the cells which go on during that animal's life, and they make new brain cells for the rats, cats and monkeys. But I was told, and I used to teach up until a few years ago, that the human brain is so complicated that we don't have that same facility. We don't have stem cells left in our adult human brain. And we don't make new brain cells. Once you get to maturity in your early, mid-20s or wherever, you've got all the brain cells for life. Well, looking in the Huntington's brains, along that margin where those stem cells lay in the animal brains, we saw it was thickened, you see, and that was right next door to the basal ganglia. And I talked to one of my colleagues, Mike Dragonoff, and I said, Mike, look, this layer, this area is thicker. Maybe there are stem cells there in the human brain, and maybe what we're seeing with this thickened layer, maybe that's new brain cells. He said, garbage. But he said, well, let's have a look. So we use very special antibodies which identify and stain up dividing brain cells. And so what we found, in fact, was that they were young, new brain cells. It took us a long time to sort this out, 5 years or so. And we had got PhD students studying it. And it became very strong. We found in all our Huntington's brains, where the basal ganglia cells right next door were dying in large numbers, these were trying to make new brain cells. Now, they clearly weren't making enough or fast enough, but they were making new brain cells. And these are people in their 50s, 60s, 70s when they died. So we got irrefutable use that evidence we were making new brain cells. So we thought we'd publish this, right? Because this turned the whole dogma around, because there's a total dogma that the human brain, that you never make any new brain cells in your adult life. And that all you're going to do is, as you get older, you're going to lose brain cells and get dumber and dumber, And so we set it off. We put it all together, this information and the science we'd done on it. We sent it off to Nature, there's two top journals in the world. One's Nature, and that's a UK journal. And the other top journal is Science, that's the United States. So we sent it off to Nature first. Went out for referees. Well, the referees were top people in the world of stem cells, and they wrote back and said, listen, these Kiwis are dreaming. And they said, unless they can actually take those cells into the lab and keep them growing and show us, we will not believe it. So it was thrown out, absolutely thrown out. So we thought, oh well, stuff it. So we did more studies, put a bit more information together, and we sent it to the other journal, Science, in the United States. And they went out to different referees in the United States, top people in the stem cell world. And they raised a few questions, they came back, but the three new referees different, obviously different to the ones for nature. They came back and said, well, this is groundbreaking work because there's a lot more work to do to really show this conclusively, but the evidence is very strong that yes, the human brain. So we got it published in Science, we got it on the front cover, and it was incredible. And and suddenly, you know, our research was becoming world famous because we turned around the whole dogma. Now, one particular lab in the United States, which would put a lot of money into this, when he read this, he actually appealed to the journal and says, I don't believe these Kiwis, and I want them to answer these questions. And the editor of Science wrote to him, he said, this is, your findings have been questioned. Can you actually refute these findings? So then we actually looked and examined, and we wrote what's called a rebuttal, and we brought more information out where we couldn't publish in the first paper because they were very restrictive on how many words you could have. So we were able to bring more of our information out, put it in the rebuttal, and get a second paper in Science which refuted them. And so you know, it just shows Kiwis, you know, we can turn the world around. So that's probably one of our top new findings, which was incredible. We used special techniques to do that. Another development which was very interesting was that we developed over a period of time, the pharmacologists, we'd been growing cells from the rat brain. We keep rat brain cells alive in the lab. And so because we're getting our human brain bequests quite soon after death, then we believe there's still some of those cells would still have some viability if we could separate them and see if we could culture and keep them alive, just individual cells alive in the lab. So over a 10-year period, with support from a philanthropic group called the Hugh Green Group, they gave us money to see if we could keep human brain cells alive. We weren't keeping the brain alive, but just separate the cells using special enzymes and just keep them alive in a dish and see if we could do research on cells in the dish from people who had died with Parkinson's, Huntington's or whatever. And over that 10-year period, the answer was yes, we could. So we were able, because we were receiving brain with a short post-mortem time, no one else in the world, we have been the first in the world to be able to grow and keep brain cells. And then you can freeze them down into what we call liquid nitrogen, which is minus 270 degrees, and then you can warm it up and then the cells will come alive, move, and then you can do more studies. Now it's not making new brains, it's looking at the brains and saying, what's wrong with the brains of a person with Parkinson's disease or Huntington's disease or epilepsy? See, so that's been a major new development, which has put us on the map. And also, you see, the wonderful thing about that is that you can grow cells from a person who's died of, say, Parkinson's or Huntington's or Alzheimer's or whatever, and you can if you keep those cells alive and you can then study and ask it questions by putting electrodes into it, or if it's from a tumour, you can put chemicals onto it and see if you can stop the tumour cells from multiplying. So we were developing a technique of having a human brain in a dish, you see what I mean? And that was incredible. So I could go on, but those are probably two of our major developments which we've had. And again, it relates back to the fact that we have a human brain bank, community and the family that are so committed to making sure we could do the best science by making sure we got the brain soon after death, by making sure that we knew all the history and so on. So you see, one of the reasons for being here tonight is to say, thank you to the community, because all the people of Aotearoa New Zealand, and for the incredible support. And it just goes to show that if you work together as teams, you know, teams within scientists, brain scientists working across the university and working with the neurologists and neurosurgeons, but most of all working with the community, I mean, which is the people of Aotearoa. Then you can move mountains. In a small country, you can actually move bigger mountains quicker than a big country. So in a very interesting way, we've put Aotearoa, New Zealand, sort of on the brain world map. And we've been able to attract people from overseas. We've just got we're just getting a new appointment, which we've appointed about 2 years ago, a world expert on motor neurone disease working in London. Just happens to be a neurologist who trained in New Zealand. He's a Kiwi. Went over there and developed, over 25 years, the world leading research group looking at the genes that are involved with motor neurone disease. Well, he's now coming back, and this year he's going to spend half his time here, next year half the time, and then after that, 0.8. And so we're developing, literally growing the lab as we speak now over the next 2 or 3 years. But he's going to keep 1 foot back in London, what's equivalent to what we call 0.2, which is 1 day a week, but it'll be in packages, so that we will have access to the funding over there. He can still keep a research lab there going, but he'll do a lot of his research here in our Centre for Brain Research, University of Auckland, and he'll bring his clinical trials here, which will help, which New Zealanders will be able to become involved with. So, and one of the things he's looking at genes, you know, looking at how we can, because in a small group of people with motor neurone disease, about 10%, there are specific genes which we do know go wrong. And so he's developing a technique whereby we can do what we call gene therapy. Try and inject into the brain special gene therapy agents, which, and I don't want to get too technical here, but we call them antisense oligonucleotides, but the little wee chemicals, and you attach a virus to it, and you put that virus, it's a non-toxic virus, but it's a virus which when it gets into the brain, we know it will multiply and make more of that little gene modifier which is attached to it, and that gene modifier attaches to the DNA and turns that bad gene off or modifies it. And so this is, and it just developed the technique. You can't just inject it into the brain, you can't put it into the spinal cord. We've done trials for that about injecting it into the cerebrospinal fluid, but if you go in under the little membrane that covers all of the brain and spinal cord called the pia mater, you just go underneath there, there's initial trials on animals look as if that will work. So I've covered quite a lot of ground there. So that's incredible. And it's ingenious. And the other thing is that will be applicable not just to the principle of trying to turn bad genes off, will be applicable not to any genetic brain disease, you see. Motor neurone disease, some of Parkinson's disease is caused by a gene, some of Alzheimer's is, and so on, and there's other rare diseases. So the principle can be modified. And so just one other thing I'll mention, which sort of follows on from that. So another thing which we have been involved with, which our human brain studies have put us in a strong position, is that we have worked with a big research group in Adelaide called the South Australian Research Development Institute. It's called SARDI, S-A-R-D-I. It's attached to a university, but it's an institute which has a big farm just north of Adelaide. And any of you who love a nice fine wine from the Barossa Valley, well, this This farm is right next door to the Barossa Valley, which is really good. You go and visit them when you're talking, just a little darts. Anyway, so they have sheep. They've got a technique where they breed sheep, and it's a huge farm, 6,000 acres, and then they use those sheep to put in genes which are taken from humans with a particular disease, with any type of genetic disease. You can put that gene into the sheep, and then you can use those sheep. It sounds simple, but it takes a long time to do this. And then you use those sheep to see if they get some of the early symptoms of the disease you're looking at, such as Huntington's or Alzheimer's, which we're working on with them. And if they do, then you can trial gene agents which you can inject into their brain and see if that is going to have an effect to stop the symptoms develop, and that will lead to a human trial. Okay, so there we are.
Yeah. I think mine's are sufficiently blown tonight. Just the fact that the Centre for Brain Research is attracting these amazing people who want to come and base part of their work in New Zealand shows the quality of what they're doing. And if I asked Sir Richard to explain to you everything the Centre for Brain Research is doing, we might be here past midnight. So I'd really like to just open up for questions. If anyone's got some burning questions, we have some lovely ladies that are going to come around with mics, and you can ask Sir Richard your burning questions.
I'll try.
I've got quite a loud voice, so I've been told.
So coming with the mic.
Thank you. Are these diseases hereditary, or can they be hereditary?
So the answer is yes, some can be hereditary, but many of them aren't. But hereditary insofar that it's inherited from your mum or dad. In other words, it's in the genes. But every brain disease causes genes to alter their function, right? Now that's not inherited. It just happens to do that.
Oh.
And so this gene therapy, for example, I'll give you an example. So we're becoming very interested. You know all about autism, right? Yes. So it's really coming, to be a major thing now. The incidence of autism is 1 in 38 in New Zealand. So every classroom has at least one autistic child. Now, we've had some of our gene people been working for 5 years, and they have found that by looking at children with autism, they can look at the genes, and some autism does actually run in the family, not all of it, but and you can see there's some genes doing things that they should not be doing, and you can work out what those genes are, and to do this, it means that you actually take a blood sample, and we've got the technique for studying genes now. You can, what we call, sequence every gene in that person. So each one of you here tonight, you've got 45,000 genes, right? That's how many genes are in every single cell in your brain, your body, everything. You've all got 45,000 genes. And if you get a condition, in this case we're talking about autism, then you get different patterns of, I found they are different patterns of gene dysfunction. And they have found in a couple of families, they have found that some of those genes relate to changing the chemicals in the brain, and when they work that one out, they can then give that person certain types of food or treatment which will make up for that. So, and this is a huge breakthrough. We're going to actually develop a special clinic just to look at autism, actually. and that's going to be announced later in the year. But so, and the problem is that they're getting that after the person's got symptoms, you see. If you did it earlier in life, you know when baby that's born gets a pinprick, and you look for different types of phenylketonuria, a very rare disease. You can change that disease by once you identify it, that young baby may be at risk of getting phenylketonuria. You can just give them a special amino acid, one amino acid, and that will prevent it. You see. So it may be that some autistic children fit in the same category. So this whole new line of study, it involves what we call big data. Now, big data is where you've got to have big computing power to be able to look and analyse 45,000 genes in each person. You see, and it takes you know, the way computers have developed now. We can crunch big data. So we're now at the stage of development that whereby we can use the modern computer technology to help answer and analyse what genes are going to be dysfunctional. It's at the early stages, but 5-10 years' time, this will become routine. And if you can work out what the gene dysfunction was in people who in 30 years' time may get Alzheimer's or may get this or that, then you can actually give treatment in advance of getting any symptoms. So the best way, one thing is that we know is that once brain cells have died, although we do make new brain cells, we never make enough to cure a disease. But if you could work out that this person is maybe at risk of this or that, and you have an effective treatment which they can take early in life, which will stop losing their cells, you know, then, so that's hope. That's hope. It's a truckload of hope, which I would never have dreamed was possible. And so, and it's coming. It's coming. That's why I mean, you know, some things in the world are, you know, progress and knowledge and computer technology is great, but sometimes you wonder, because often gets disused. And that's always the problem. So, and we see that all around with social media and all the rest of it. You've got these wonderful facilities which can actually help solve a disease, but you can also be used the wrong way. So balancing all this up is a bit of a challenge, but nevertheless, as far as brain research goes, we've got to make sure we use all that stuff. And that's why we have our bright young kids. Honestly, what they can do on computers is absolutely mind-blowing. Does that help? It's a long answer to a short question. So there's enormous hope.
I'm interested in the relevance of exposure to chemicals and also having had concussion previously to getting any of these diseases.
So there are a number of very clear chemicals herbicides, which we now know are totally toxic to the brain. You know, I remember giving a talk on Parkinson's disease some years ago. It's on the North Shore in Auckland. And there were a couple of young guys there who were, the job they did was to spray gorse. And they used very powerful herbicides. This is before the toxicity of some of these chemicals were known. And of course, they do it on a hot day. And they'd be just wearing shorts. And if it got too hot, they'd just spray themselves to cool down. Oh. Well, you can absorb through the skin these powerful chemicals. So taking care, you know, Dearest dust is another one, often chemicals which are used in the garden or whatever. And that's why we've learnt to be very careful. Your skin is an organ and you can absorb through it. And it will ingest whatever. So So yeah, our bodies are very finely balanced, and you've got to be very careful about exposure. I mean, we've heard, you know, thalidomide, that whole terrible thing of women who are exposed to taking thalidomide to help them sleep, and then they have deformed babies and so on. There's plenty of examples out there which show that toxicity in taking compounds or chemicals must be, you know, the health and safety is absolutely vital, and we've learned that through our, you know, through terrible examples. So yeah, the answer to your question is that we know that certainly in Parkinson's disease, we know certain chemicals can knock out the same cells that normally die in Parkinson and give a person drug-induced Parkinsonism. I shouldn't do a lot of talking. Yeah.
Oh, there's another question. Thank you. I'm just wondering if you've done any research on diet and the effect of diet might have on some of these conditions at all. Is there any research doing on the diet? Could that possibly help or hinder or whatever?
Yeah, there's a lot of research on diet. So what you eat, you know, and there's a lot of, so we have brain cells, they communicate with each other by releasing chemicals at the end of the nerve fibres, which then attach to the next brain cell and pass on information. So the way that the brain cells communicate is by releasing chemicals, and there's a whole range. There's probably, last count, it's probably something like 250 different very small chemicals which are used by brain cells to talk to other brain cells. And what's really interesting is that many of those chemicals can relate to particular types of food in your diet, different types of amino acids. So, and so there's a lot of work going into what food is good for your brain. And there's, I'm not an expert in it, but we've got nutritionists at the University of Auckland, but elsewhere all around the world, looking and telling you that certain types of, you know, cheeses and all sorts of different types of food have different concentrations of different chemicals, some of which often this can be fine, but different people may be allergic or may, you know, it varies between people too, which makes it, I mean, my wife's just found out that she's just discovered that a lot of her problems that she's had with eating types of, so she's ended up being dairy-free and which is terrible for me. But you know, and we've got, I know our 5 children, two of the girls they have to, you know, they've got certain restrictions on their diet, and that was found out. So food is vital. There's good food, and you just have to, well, just Google the food, you know, good foods and bad foods for your brain, and it'll bring it up. And it's actually not, it's pretty, the sort of information they have is very, very interesting. So the other thing which is, you know, not only that, but it's very clear now that, look, you got to look after your body. I mean, and every time your heart pumps, you know, it pumps, it sends blood all around your body, right? Now, your brain is so vital, it needs to get a very good blood supply. So every time, although your brain weighs, 1,500 grams, and that's 2% of your body. So it's only, your brain's only 2% of the body. But every time your heart contracts, 25 or 20 to 25% of the blood coming out of the heart on each beat goes straight to the brain. In other words, the brain is top priority for blood, critical. And so and you know, your heart is a pump, and it's got arteries around it, and it's a pretty incredibly efficient pump. So there's one good rule, and that is what is good for your heart is good for your brain, because you've got to have a good heart in order to keep a good brain. So the things that are good for your heart, well, the first thing, and I'm trying to keep my figure trim, but is obesity it's bad for your body, but bad for your heart and actually bad for your brain. So losing a bit of weight, trying to get down to your ideal, and you got to still make life worthwhile, I mean, you don't. But it's actually a very good fundamental element. So looking after your heart, exercise in moderation is actually good. I've just had my second hip. I had my left hip replaced, 10 years ago. That was fantastic. And then over a period of a few weeks, my right hip packed up and I had a hip replacement April last year. And it's taken a little while to get over that because I'm a bit older now. But I can walk now. So doing a bit of it, I like walking. I can't stand jogging because I've never seen a happy jogger. I tried going to the gym, but they all look as if they were like my rats at work, doing the same thing. But I love walking because walking you can stop and talk to people, and you can enjoy it and smell the roses, and so that's for me. But everyone, I mean, everyone's got to do so, and I feel better. So those are just a couple. And of course, smoking, despite what the government's done, right? Smoking is out. It is poison, and vaping is even worse. So if you got a good car, you look after it, you polish it, right? And that means you still want to keep using it, but don't abuse it. You wouldn't fill your car up with the wrong sort, if it's a petrol car, you're not going to fill it up with diesel, right? You're going to give it the right food. You're going to make sure it's oiled well. Keep moving. You may not be able to move as good as you could 10 years ago, but just keep moving. So I've got getting over my hip because I was actually on crutches for about 9 months, and it's taken me a while to get my muscles back. Went to see the physio. She gave me exercise, which I do, and one of the ones is crouching. Crouching is good. It's helped me get over my hip. And my muscles are pretty well back, but that's one of the best exercises, doing a bit of crouching. I also heard on the radio on the weekend that actually crouching against the wall, you would slide up and down the wall, that's supposed to be good. There's a lot of good things to do, but I always say you've got to do it in moderation. You don't, you don't have to eat carrots for the rest of your life to live, just to live another 2 or 3 years, drive me nuts, right? But so it's about balancing. And despite that, as you get older, well, we're not all going to live forever. So things are going to catch up with you, but just put quality into your life. But enjoy it. A glass of red wine, a nice red wine, is wonderful, but you don't have to drink the whole bottle, But there's a few rules which are sort of common sense, so it's a bit like what grandmother used to tell you. And if yes, there's about 10 different things, for instance, and just Google, the best, what's good for preventing Alzheimer's disease, and I'll give you a list of things and types of food. Mediterranean diet is good. Probably not having steak every night, but you got to have some steak, I believe. But just put it in the moderation, but first and foremost, you got to enjoy your life, I believe. And one of the things, one of our sons has gone to medicine, and he's working up in Whangarei Hospital, and loves it, and of course short-staffed and all the rest of it. But one of the things they have learned, they were taught, is that, when you go and see your doctor, and I tell my medical students this, and I teach them, I'm teaching on Monday, and I said, everything doesn't necessarily need a tablet, but the best tablet you can sometimes give your patients is actually your ear. Listen to their story, and be empathetic, because often they will come along And you may not be able to give them the cure, but make them feel better, give them hope. And the most powerful medicine you can sometimes give someone is just time. And we're getting a bit obsessed now because, you know, doctors, nurses, health professionals, they get overloaded, but we must never forget that the best thing you can give people to help them keep and stay healthy is time and good advice and understanding, you know, and often they'll come and, you know, I know we're also told that sometimes when a person goes to visit the doctor and they take their child along, they usually put their child, they'll present the child as being sick, you know, when really just going out the door they'll say, doctor say, and how are you feeling? And then you get the story. The person went, is it not because the child was sick, but because they've got a problem which they just couldn't put out there. Anyway, it's a bit of, I'm getting a bit random.
I think we're getting a lot of really good life advice here tonight as well. But if anyone else has any more burning questions, Sir Richard's going to stay around and join us for supper. So please do feel free to talk with him. I feel like he's already greeted everybody in the room already.
One question, final question.
Have you been looking at any brains that have been from sports persons, rugby players, league players?
So what we're doing now, we've actually, because it's become very, very clear that, you know, people in their prime years, if you get a brain injury, then it often can leave a mark in your brain which will not totally disappear. And so people with concussion, you know, the old concussion you can fully recover from, but when it starts, we now know there's strong evidence that multiple concussions or injuries can be a predisposing factor to your brain health later in life. Often may lead to dementia. One of the risk factors for dementia is brain injury earlier in your life. Now, it doesn't mean to say that, you know, obviously no injury is the best thing, but, you know, we do get bangs on the head, and no matter what sport you're in or whatever it happens, it does leave a mark on your brain, and often that will continue as a little area of what we call inflammation in the brain. And so we have started what's called a sports brain bank, part of the Neurological Foundation brain bank. We're collecting the brains of people who have had some All Blacks or other people who have had injuries during life and get dementia later in life, and they're donating the brains to us to see if we can work out what is actually going on. And we're working with people in Australia and also a big group in the United States that multiple injuries to the brain result in a type of quite characteristic brain pathology, not dissimilar to dementia, excepting it's at the bottom of all the folds in the brain. They go in and out like that, which is wonderful. But when a person gets an injury, often it's the cells at the bottom of those folds which seem to get more affected, and you can see the typical bad amyloid accumulation at the bottom of those cells, which are characteristic of long-term multiple injuries of the brain. So we're trying to work out to test this, and then, I mean, you know, we don't want to say don't do sport, but we want to say do sport as safe as you can. And there's an awareness now, slowly coming in, that we've got to try and make our sport as safe as we can. So the thing, like what we can say is that getting a bang on the head does not improve your intelligence, that's for sure. And getting too many of them is not good for you, but we need to work out how to do that to make life still interesting and still make sport a challenge, you know, but do it safe. Thank you.
Yeah, and if you want to keep up with what's happening in the world of neuroscience and the breakthroughs that are happening, the Neurological Foundation does a free quarterly magazine. It was on your chair tonight, and there'll be information on there if you want to sign up for that free magazine. If you're in a position to make a donation tonight, our lovely ladies at the back have some envelopes and donation slips as well. Or if you just want to ask any of the ladies from the Neurological Foundation any more about the foundation or doing events or fundraising, anything like that, just have a chat to them tonight. But I just really want to say thanks for coming, and it's been such an engaging event. I've loved it, and I hope that you have too, and I hope you can also stay around and join us for a cup of tea and a bite to eat tonight.
Great.
Thank you.
Thank you very much. What's the time? Yeah. Thank you. Thank you.